9. It's much easier than starting at 9 where half the class comes in 10 minutes late. So in today's class we're going to move away from artificial networks for deep learning, to look at the structures that have inspired them and inspired their development, which is to say networks of biological neurons learning about our environment to guide our behavior. So I know that many of you found the last class a little simple because you come from a computer science background, and for you guys this will probably be relatively challenging, whereas for those of you that come from a cognitive and biological background this will probably be a bit easier, and the last class will have been a bit more challenging. And indeed it's like that all the way through the course, right? If you're a good coder, you'll find the coding parts of the lab exercises easy enough, but the other parts you'll find a bit harder, and it's not just an exercise about coding. Whereas if you come from a cognitive science background with less coding experience, you'll find the cognitive parts a bit easier and the coding parts maybe a bit harder. Okay. So why do we think it's important to study biological networks? Well, first of all, these are the inspiration for many machine learning networks in artificial intelligence, including deep convolutional neural networks. Second, AI networks typically simplify indeed always simplify the processes involved here for efficiency, right? So there's a lot of mathematical abstractions in artificial neural networks to make things faster and more efficient and generally make things work, right? But biological networks don't work in exactly the same way. And AI deep networks are not yet as advanced as the human brain, partly because they're making lots of shortcuts for computational efficiency, and even on modern computers, the artificial deep networks are really pretty demanding for computers, right? So another reason is that where the aim of deep learning is to imitate human behavior, which is one aim of deep learning, certainly not the only aim of deep learning. Sometimes it's just used for engineering. But where the aim is to imitate human behavior, for example, in tagging images on the internet, which is something that humans have traditionally done, right? Following neural mechanisms more closely may help to more closely imitate human behavior than using the mathematical abstractions that result from engineering in computer science. The last point is really kind of a theoretical point. So biological deep learning is really the major link between, or one of the major links between AI and other sciences. Okay, so you'll see here that what I'm trying to do here is to link artificial intelligence to biological and cognitive sciences, rather than, as computer science would teach it, which is to link it to maths and computer science. Right? So that's kind of a different angle on things. Right? And we think that this kind of has the potential to draw more of a close relationship between these different sciences, and so broaden your education. But definitely for the more cognitive among you, there's traditionally been a big gulf between the biological. Okay, so we understand the physical sciences in terms of mathematics perfectly well, right? We understand chemistry in terms of physics perfectly well. We understand biology in terms of chemistry perfectly well, right? But the social sciences we do not understand in terms of the other sciences. The social sciences do not easily link to biology. Right? But if you understand human behavior as being a result of biology, and that's kind of what we're aiming to do with this, then it brings the social sciences into a link with other sciences, right? So with biological sciences as well as with computer science. Okay. So because you guys have some very different abilities and backgrounds, we're going to start really from the start here. So we're going to start with what is a neuron, right? Okay. So a neuron, all right, is a specialized type of animal cell. Okay. Most aspects of neuron biology work just like other cells and aren't important for performing computations. So for our class, are not important at all. All right. But the cell membrane in neurons, which is this thing going around the edge of the neurons here, okay, is very unusual. in terms of animal cells. Most animal cells are more round than this. All right, this is very much spiky and elongated. All right, and it has a very extended membrane, so a very large surface area compared to most cells, because the membrane is where all the action is happening computationally. Okay. So this cell membrane is specialized to perform simple computations using electrical activity. Right. So it's kind of the biological equivalent of kind of everything that's going on in computer science as well. So first we have a group of dendrites, which are these things at the end here, and these are integrating incoming signals from lots of different neurons. There therefore are lots of neurons that this neuron is connected to through these dendrites here. These connections between these dendrites and all of the other neurons that they're connected to vary in strength, and they change the strength of the connection between neurons, and therefore how well a signal is passed between neurons, depending on past activity. Okay. So they're the biological equivalent of a convolutional filter. Right? And changes in synapse structure, which is to say the links between different neurons at the dendrites, are how we learn. Right? So they're kind of the biological equivalent of machine learning. Okay. If these incoming signals from the dendrites are strong enough to pass a particular threshold, which is a concept that we've seen before, right? Then this neuron will send an electrical impulse down this axon, which is more or less just a long wire, okay, and then it will pass that signal on to other neurons that are connected at the axon terminals at this end here. Okay? So that would be the next layer in a deep convolution on that one. Okay, so the basic component of a biological neuron's membrane is a thing called an ion channel. Okay? And this can allow charged atoms, or ions, primarily of sodium and potassium, that is to say natrium and kalium in Dutch, to pass into and out of the neuron. Right? But these ion channels are normally closed. Right? So they block ion passage. And these ion channels are also embedded in a cell membrane, which is this structure here. Right? And this also doesn't allow ions to pass through. So in the closed state, this whole membrane just doesn't allow ions to pass through. So because these ions can't pass through the membrane, this allows the cell membrane to have an imbalance of ions. There's a lot more sodium ions outside the cell membrane and a lot more potassium ions inside the cell membrane. There's another state of the ion channel which is an open state, and this does allow these ions to pass through, and only one particular type of ion. So in this case, this is a sodium ion channel, so it will allow sodium ions to pass through, but will not allow potassium ions to pass through in the other direction. So, because these are electrically charged, right, this is just labeling the different parts, and this is an actual what the protein actually looks like. See here. Yeah, because these are electrically charged atoms, when they cross the cell membrane, right, they change the electrical potential or voltage across the cell membrane. Okay, and that's called the membrane potential, the electrical voltage across the membrane. And so one important thing to notice here is that this is a passive mechanism. It's just a hole that can open and close. Right? It doesn't pump ions against their concentration gradient. Passing through an ion channel, the ions can only go in the direction from more ions to less ions. Okay? So it doesn't allow ions to kind of diffuse against their concentration gradient, as we would call it. Because this ion channel is passive, therefore, it only allows ions to move towards lower concentrations or sometimes to overcome voltage differences. And this is a very fast mechanism because it's passive. Right? It's much faster than actively pumping ions around is. But the ion imbalance that all of this relies on needs to be established in order to do this. Okay? And so there's a second mechanism that's doing this, establishing this imbalance of ions, and this is known as a sodium-potassium pump, or sodium-potassium ATPase, because it's basically an enzyme that breaks down ATP, which is the biological energy currency. So this pump protein here is also embedded in the ion channel. And because there's more sodium outside of the cell, all right, and more potassium inside of the cell, it's working against the concentration gradient. All right? So what it's doing is it's getting the small amount of sodium that's inside the cell and putting it outside of the cell, small amount of potassium that's outside of the cell, putting it inside the cell. All right? So it's establishing this difference in ion concentrations across the cell membrane. All right? And this allows the cell membrane to reach this kind of imbalanced rest state, okay, which is known as the resting potential. And this is a voltage of around minus 70 millivolts. And if there was no active pumping at all, there would just be completely even distribution across the cell membrane. So this is a very, very important structure for establishing this imbalance of ions that everything else will rely on. Okay. So I know that's a little bit biological and technical for some of you, so we're just going to go again through that basic bit again. Okay. So cell membranes of neurons are specialized for electrical computations. Ions are atoms with electrical charges. Cell membranes do not allow ions through. But sodium and potassium pumps establish an imbalance of ions across the cell membrane with much more sodium outside and a little more potassium inside. Then opening an ion channel allows ions to pass through in the opposite direction towards a more balanced concentration across the cell membrane. So, how do these ion channels open? Right. So, opening a sodium ion channel causes sodium to enter the cell through the cell membrane, right, changing the membrane voltage or membrane potential, as it's often called in biology. So this may lead the cell to activate or fire if the amount of activation is what's simulated by the pixel values in feature maps of an artificial convolutional neural network. For our purposes, there are two important ways that an ion channel can open. All right. First of all, it can open because a neurotransmitter from a different neuron binds to the ion channel, changing its shape, right, and causing the ion channel to open, and therefore allow sodium ions to enter the cell. Right. So a neurotransmitter is a signal, a chemical signal, released by presynaptic neurons, or a neuron before the synapse on the dendrite. Okay. And so this will potentially cause activation of the next layer of the network of neurons. So here we're going to look at, here we're going to look at activation of the ion channel by a particular chemical called glutamate, which is the most common neurotransmitter. And so what normally, the main way of thinking about this is that when glutamate is released from this presynaptic terminal here, this crosses the synaptic gap, or synaptic cleft as it's called, binds to this postsynaptic ion channel, therefore allows sodium to enter and moves the membrane potential more towards 0 from minus 70, its resting state. There are also neurotransmitters that kind of do the opposite. All right, so that will further increase the membrane potential, so hyperpolarize the cell, as we call that. So gamma aminobutyric acid, this chemical here, which is very similar to glutamate, is the most common inhibitory neurotransmitter. So when GABA binds to the postsynaptic cleft, okay, that reduces the activity of the postsynaptic cell. Okay. So basically, we've got two different molecules that can push the postsynaptic cell's activity in two different directions. Glutamate excites the postsynaptic cell. GABA inhibits the postsynaptic cell. Other neurotransmitters work by similar mechanisms often, but glutamate and GABA are by far the most common in mammalian systems. So the second way that an ion channel can open is due to a change in membrane voltage. All right. So this is particularly important because opening the ion channel will also change membrane voltage itself. All right. So changing membrane voltage, right, can lead to further changes in membrane voltage by activating this second type of ion channel. So in this way then, a voltage-gated ion channel acts much like an electronic transistor, which is the electrical switch that opens and closes as a result of electrical input, which is the basis of all computer processors. Right? So what a voltage-gated ion channel does is, when the membrane voltage reaches a particular point, okay, that causes the ion channel to go from a closed state here, okay, to an open state due to a change in the potential across the membrane, the voltage across the membrane here. Okay. And that causes more sodium to enter the cell. Okay, so it causes a larger change in membrane potential. You can think of this as kind of amplifying the signal, but it's a very, very large effect, this, because there's a lot of voltage-gated sodium channels in biological neurons. Okay. So what that does is it basically does the threshold operation. Okay. So if we have one neurotransmitter binding, okay, an excitatory neurotransmitter that pushes the membrane potential sort of closer towards zero, so it starts in this very strongly negative state. Then, if it doesn't reach the threshold that activates the voltage-gated sodium channel, then this signal, this voltage then just drops back to rest again as the pumping mechanism sort of moves the ions back around. But if the incoming signal is strong enough to reach this particular activation threshold here, okay, so for example by several neurotransmitter molecules binding, right, then this will reach a particular threshold that causes the voltage-gated sodium channels to open, right? And that causes a very, very large change in membrane potential, known as an action potential. So the threshold for activation of the voltage-gated sodium channel is the biological equivalent of the threshold or rectify operation we saw in artificial networks. So if there's some incoming signal but it's not very strong, right, nothing happens to the postsynaptic cell. Right? But if that reaches a particular threshold, a lot happens. Right? So then the cell starts firing, it becomes very active. So until, just like we saw in the threshold operation, until we reach a certain threshold voltage, there's going to be no response. Right? And in biological neurons, this threshold is not zero, right? But that doesn't really matter. Depending on the type of cell, is minus 55 or minus 15 millivolts, because there's an ion imbalance of ions maintained at rest. Right? So there's always an imbalance of ions across the cell membrane. So zero doesn't really have any particular meaning here. If the incoming signal does exceed that threshold, then the stronger the signal is, the stronger the firing rate of the postsynaptic neuron is. Okay, so then the strength of the output scales with the strength of the input after we reach this threshold. Okay, so this is very much like the threshold we saw in artificial neural networks. Okay, and at some point, the neuron will reach a maximum firing rate where it just can't fire any more fast than that. Okay. So then we also have a maximum activation in all biological neurons. Yeah. Yeah, it's a limitation of the biological system. The reason for that maximum firing rate, so the maximum amount. So yeah. An important feature of the activity of neurons is that they fire in discrete packets, right? So they're on or off, right? The amount of activation, therefore, is the firing rate. So how quickly they're firing, how quickly they're coming on and off, right? Okay. So the reason why we have this maximum firing rate is because the action potential takes time. All right. It's not very much time. You can see here it's a couple of milliseconds. All right. But it does actually take time. Okay. So if we therefore get a bunch of, we can see here a bunch of failed initiations, which are incoming signals that aren't strong enough, right? But if we get incoming signals that reach this threshold, then we have this action potential, which is caused by the opening of voltage-gated sodium channels, which then reaches a peak after a certain time, then drops back down, and eventually comes back to rest. Okay? And this establishing rest again takes some time because the ions need to be pumped around again. So the high voltage that we see here causes the sodium channel to close again. All right? But this is a different closed state than before. So the channel protein has a different closed shape. Indeed, we can see this a couple of slides ago. Here we can see the initial closed state of the voltage-gated sodium channel. Okay? Then after a particular threshold is reached, we see this opens, right? And then it closes again, but this state and this state are different. Yeah?
But if it's a limitation, would you then want to hard-code it in your neural network?
That's a good question. If you want your neural network to act more like a biological system, then you would want to put it in, right? But it's evolved to be there, right? So whether it's there for a good reason or not depends on the hand of God, if you like to put it that way, right? So that's just how biological systems work. It's not good or bad, it just is, right? It's how it evolved, and evolution is messy. So it has effects. Whether we consider those effects good or bad is very, it's putting a human spin on things, right? Yeah. So, then this initial repolarization, so this falling phase that we see here, okay, relies on potassium channels opening, which allows potassium to exit the cell, so go in the opposite direction than sodium entering the cell then. Okay? So that causes the membrane potential to fall again, and indeed to go a little bit below the resting potential. But then the imbalance of ions across the membrane is different than it is in the resting state. So what we see here in this undershoot period is this resting potential being re-established by pumping those ions back around, which is why this all takes time and why we've got a maximum firing rate here. Activity in a presynaptic neuron can also inhibit the activity of postsynaptic cells by causing the membrane to further polarize, okay, making the membrane voltage more negative. Okay. The GABA receptor, which is what we see here, is also an ion channel, but it's an ion channel that allows negatively charged ions, chloride ions, to enter the cell. Okay? So it pushes the cell membrane potential in the opposite direction, right, to sodium ions entering the cell. So when GABA binds, or when alcohol binds, or when benzodiazepines like Valium bind to this channel, then it causes chloride ions to enter the cell, and therefore it makes the cell less likely to fire. Okay, this pushes the cell further away from activation. Okay. So we have basically both positive and negative inputs, known as excitatory postsynaptic potentials, or EPSPs, and inhibitory postsynaptic potentials, IPSPs. Okay. And these can come from typically come from different presynaptic neurons. So here we can see an excitatory neuron in red and an inhibitory neuron in blue. Okay? And these are both going to affect the membrane potential. So the excitatory one is going to push it up a little bit towards zero. The inhibitory one is going to push it down a little bit away from zero. And so if we look at how that works from a bunch of inputs from different places, so here we've got in green here we've got excitatory inputs, and in red we've got inhibitory inputs. Okay, so if we start at the resting membrane potential here, we can have a couple of excitatory inputs coming in, and so pushing things towards the threshold here, and then an inhibitory input coming in, pushing things away from the threshold, and then that's going to drop away again because nothing happens for a moment. Then the more excitatory inputs come in, and then more inhibitory inputs. And then eventually an excitatory input comes in that's strong enough to reach the membrane potential here, the threshold potential here, which will cause the cell to fire and have an action potential, which is that process we've just seen there. Yeah?
How come this is happening at the red square and not at the start of
Yeah. So in the right. So it was. This is a detail that you do not need to know at all. It was until very recently thought that all of this summation occurs at this point here, which is the axon hillock, the start of the axon where the action potential initiates. But more recently, it's been determined that there are more local effects here. So if you have an inhibitory and an excitatory input coming in on the same part of the dendritic tree, they will interact there. Right? So this is kind of the simple way of doing it, saying, well, the only point that really matters is this point here, okay, the axon hillock. But actually, there are smaller effects occurring everywhere around the dendritic tree. So these inputs can come from a range of different places, right? But there's also a limited spatial distribution of those inputs, all right? Because the dendritic tree only has a certain size. And we also see that there are negative and positive inputs here, right? Spread over this tree, and if we reach a particular threshold, this will cause firing, right? So this might seem quite familiar because in a convolutional network, the filters imitate this structure. They have positive and negative filter values, which are like positive and negative inputs to a neuron, that depend on the activation of the previous layer, which in this case we're going to see these as the activations of previous neurons in the earlier layer. Okay? And so the activation of the next layer's neuron, so this is the neuron that we're actually looking at here, okay, will depend on these synaptic strengths multiplied by these activities of the presynaptic neuron. These synaptic strengths can be both negative and positive. And then if this reaches a particular threshold, as we saw, this causes the postsynaptic cell to fire. And if it's below that threshold, as it is in this case, right, then the response to the postsynaptic cell will just return to zero. Yeah?
I'm kind of missing the weight sharing over the image.
Yeah, there aren't shared weights. We're going to come to that later. Yeah, there aren't shared weights still. So, to summarize that, neurotransmitters, primarily GABA and glutamate, released from a presynaptic neuron, can excite or depolarize or inhibit, hyperpolarize, activity in the postsynaptic neuron. Okay. This relies on ligand-gated ion channels, that is, say, neurotransmitter-activated ion channels. If the membrane polarization reaches a particular threshold, then voltage-gated ion channels open. This results from having many excitatory inputs coming in a short period of time. This will then strongly depolarize the neuron, producing a thing called an action potential. Once the action potential has been triggered, it travels from the kind of input end, the dendritic tree end of the neuron, down this thing here, the axon, right, to the sort of output end of the neuron, okay, which will then synapse with the dendrites of another bunch of neurons. One feature of this is that this axon here can be very long. All right, so it can go from one end of the brain to the other quite easily. It can also go from the top of the head to the feet. All right, so these can be very, very long fibers here. So this allows kind of transference of a signal from one place in the brain to somewhere very different as well. Once an action potential has been triggered, so this will pass then, the action potential will then pass along this axon basically unimpeded. Nothing really happens in the axon. It's more or less just carrying the signal from one end to the other. And this process also relies on voltage-gated sodium channels. Okay? So basically what happens here is if we have a activation of the voltage-gated sodium channels at one location here, this will cause sodium to enter the cell membrane. Right? And then this will change the voltage on this part of the cell membrane. Right? Okay, so then on this part of the cell membrane, the voltage-gated sodium channels will then become activated. Right? And then this will cause more sodium to enter in this location, right? Activating these voltage-gated sodium channels. And then more sodium comes in here, and then activates these ones as well. Okay, so it basically just allows the signal to be passed along the wire here. So basically what's happening is that each of these locations where the sodium comes in, it causes the nearby locations to also activate their ion channels and cause more sodium to come in. And so there's this kind of cascade passing along the axon. Okay. Kind of spreads like a wave down the axon. You may think, if you pay attention to this, that this would also activate ion channels here, so backwards down the axon as well, right? Activating those. But as we've already seen, after the ion channel's been activated, it returns to a different state, which doesn't allow it to be activated again. Right? So this means that the signal can only go from activated place to a not previously activated place and can't go back to a previously activated place. On reaching the end of the axon, a presynaptic terminal, so that is to say that before the synapse to the next neuron, the action potential then leads to neurotransmitter release. Okay? So once the signal reaches here, what happens is a different group of voltage-gated ion channels opens. This is particularly important to know, but these are calcium channels, which cause calcium to enter the cell, and we've got little bubbles of neurotransmitter here, right? And these are suspended in proteins. When calcium enters the cell, the proteins change shape, which draws these little bubbles of neurotransmitter onto the synaptic cleft here, releasing the neurotransmitter, and then beginning the cycle again in the next neuron. Okay. So, right. We're going to come on to shared weights in a minute, but how are the weights established in a system like this? All right. Backpropagation isn't going to work in a system like this. All right. So a completely different system is involved here. Okay. So connection weights in biological systems rarely depend on anything that looks like backpropagation of error. Okay. And this is important to realize because humans don't learn about the world through supervised methods. All right. When we're exploring the world as babies, we don't learn that we're doing the wrong thing by having somebody tell us that we're doing the wrong thing. All right? So the way we learn about the world instead, there's no correct response in learning about the world. Right? Well, we'll come to how we actually learn about the world in a minute. However, there can be adaptive and maladaptive responses and behaviours. That is to say, responses that will help us survive, adaptive responses, and responses that make it harder to survive, maladaptive responses. So we can actually cause something that looks quite a lot like backpropagation of error using a device called a Skinner box. So this is an apparatus used in animal experiments that delivers rewards, food, right, for good behaviours of the animal, okay? And it delivers punishments, electric shocks through a grid on the floor, for bad behaviours of the animal. So this allows you to watch what the animal is doing, right, and then train it to do what you want it to do. Right? Rather a cruel method, but there you go. These are known as conditioned responses because the animal makes a behaviour of its own choosing, right? And then it's either rewarded for that behaviour or punished for that behaviour, and that determines what it will do in future, all right, to seek reward and avoid punishment. So conditioned responses like these, learned from punishments and rewards, however, are unlikely to. So this basically tells the animal, was your behaviour right or was it wrong? So it gives the animal an error signal, much like backpropagation of error, right? But conditioned responses like this are unlikely to work through backpropagation-like mechanisms because in backpropagation we're affecting the structure of the whole network, right? But here it's much more likely that the animal will change its behaviour based on a single decision stage at the end, all right? So it's going to still perceive the world in the same way, but it will make decisions in a different way. So you're really only affecting one later stage in the processing here. So this doesn't really work like backpropagation of error at all. So instead, we learn by unsupervised processes. And specifically, we learn to recognize patterns of activity that we've seen before. Right? So effectively what we're doing is we're learning the statistics of the world that we live in. We learn what patterns are common in the world, what patterns are uncommon in the world. what patterns commonly lead to food, what patterns commonly don't lead to food, etc. Right? So this is done through a mechanism called long-term potentiation that we're going to look at in a second. And this is basically a lasting enhancement of synaptic connections by co-activation of presynaptic and postsynaptic neurons. That is to say, if the activation of the presynaptic neuron normally leads to activation of the postsynaptic neuron, right, then we get a stronger connection between the two neurons. This is often described by Hebb's postulate, which is cells that fire together, wire together. That is to say, cells that are activated at the same time, right, generally form strong connections between each other. Another feature of, a related feature of biological neuron is that cells that wire together, cells that form strong connections with each other, typically also lie together. That is to say, they're normally found in nearby places. This is known as the minimal wiring hypothesis. And the goal here is to reduce the required connection length between neurons by getting neurons that normally need to communicate with each other and putting them near each other so that they can communicate more easily. And this, of course, increases efficiency, reduces the amount of wiring you need, increases the speed of processing, and so on. Okay. So this is not particularly important at the moment, but it's a very close relationship to this cells that fire together, wire together, is that cells that fire together, wire together, and then that will often be cells that are close together. All right, so how does neural activity lead to a change in synaptic strength? Right, so how does cells firing together lead to cells wiring together? So there's a lot of mechanisms that do this. We're just going to walk through one because it kind of illustrates the main points of what's going on here. But postsynaptic neural activity primarily modifies synapse structure at both the presynaptic terminal. So if this cell, the postsynaptic cell, is activated, it modifies the structure of this terminal and also of this terminal. Okay, so it forms stronger connections at both ends here. So increases in presynaptic neurotransmitter concentration and release are one mechanism. So basically, well, let's just go through this step by step. It's probably the easiest thing to do. So basically, what we've got here is we've got a presynaptic neuron releasing some neurotransmitter, right? This leads to sodium entry into the postsynaptic cell, which leads to excitatory postsynaptic potentials happening at this time at this place. Okay. Added together with other excitatory postsynaptic potentials from the rest of the dendritic tree, this may cause enough depolarization to make the cell fire, okay, but also enough depolarization to force a magnesium ion, which sits in this type of receptor here, okay, out of the receptor. And when that happens, okay, then calcium ions can enter, okay, which causes a bunch of effects that basically increase the expression of this protein here. Okay. So to go through that again, when sodium comes in, all right, if this then leads to the cell firing, all right, then it causes calcium also to enter the cell, which increases the expression and the availability of this receptor here. Okay. So basically then that makes more receptors for these ion channels, so it'll make the connection stronger. Yeah.
So the weights are actually adaptive, they can actually change over the life.
Yeah. There are short-term mechanisms, which this is a relatively short-term mechanism. There are much longer-term mechanisms as well. So another effect of calcium entering here is that the postsynaptic cell then releases a different chemical called nitric oxide, which diffuses back to the presynaptic cell and increases the amount of glutamate release and the amount of glutamate production. Okay? So there it's changing the activity at the presynaptic cell end as well. Another effect of calcium entry and cell depolarization is that the electrical activity of neurons causes changes in their gene expression as well. So this is a much longer lasting effect, right, where more of these ion channel proteins will be built, right, by the cell nucleus. So basically more genes will be expressed to make more and more of these. So again, increasing the strength of this connection. Right? So there are lots of different mechanisms going on there that happen at different timescales. The simplest mechanism here of just making this receptor active instead of inactive is a very, very short-term mechanism. Right? Increasing the availability of this is kind of a medium-term mechanism. Right? And increasing the expression of this, so making more of these protein, is a longer-term mechanism. Right? So there's lots of different effects going at different rates here. Okay. So we've looked at, so I should also mention here, because people generally find this interesting, several common drugs interfere with these processes. Okay? So these processes, basically changing the strength of the connections, is the basis of memory. All right? It's the basis of learning and memory. So various drugs, alcohol and benzodiazepines, like Valium, reduce the spiking activity of the postsynaptic cell because they activate GABA receptors, as we saw earlier. So this reduces the probability of depolarization. Right? So it reduces the ability of the cell to change its membrane structure. Right? Therefore, it reduces our ability to form new memories, which is why when you're messed up on alcohol or Valium, you do not remember what happened. Right? Ketamine, another fairly common drug of abuse, blocks the activity of this receptor here, the NMDA receptor. Okay, and this is a vital step in strengthening all of these weights. Okay, so all of these processes rely on this activity. So if we block activity at this receptor using ketamine, then we basically can't form new memories at all. Right, so we find it very, very difficult to have any type of strengthening of the synapse if ketamine is in the bloodstream. Okay, so the story so far, we've seen that. Okay, so how does all of this, just to remind you how all of this relates to the systems we were looking at in the last lecture, okay? Network layers in biological networks are layers of neurons at different levels of synapses. That's to say, from one synapses away from the retina to two synapses away from the retina, three synapses away, and so on. Okay. Feature map, we've kind of ignored so far. We're going to look at that in a minute. Okay. A filter basically is a result of is expressed as the integration of excitatory postsynaptic potentials and inhibitory postsynaptic potentials across the tree of dendrites. Okay. A threshold is the activation point at which voltage-gated sodium channels are activated by above threshold depolarization by strong excitatory postsynaptic potentials. Pooling operations, well, they don't exist, but we'll look at them in a minute. Normalization operations, we also haven't looked at. Okay. The learning mechanism is unsupervised learning. It's Hebbian learning. Cells that fire together wire together. And this results from activity-dependent changes in synapse structure and strength. Okay, that is to say, a change in synaptic weights. Jacobus.
So I can see how the structure changes to increase the connection weight and then you can block to decrease the time.
Yeah.
Can you also decrease the connection weight?
There are mechanisms that do that, but they primarily happen during childhood. So during childhood, connection strengths are continuously decreasing. So as we're learning about the world, connection strengths, if you're doing nothing with a neural circuit, just keep decreasing. Right? So that's a process called pruning. It's one of the things that goes wrong in autism. If you are continuously decreasing the connection weights, then you need activity to keep them stable. All right? So there are processes like that, but inhibition of activity is often just specific for a particular moment, so it doesn't generally cause a decrease in connection weights.
So does that mean that everything's constantly growing, so you need more transmitters to eventually?
Yeah, so there's just, even after childhood, there is a slow decrease in synapse structure. Yeah.
Are there neurons requirements that use Hebbian learning that we use in production?
Sorry, again?
Are there methods, algorithms that use Hebbian learning
Yeah.
that we use in production?
Yeah, so unsupervised machine learning generally uses Hebbian learning-like processes, right? Just because it's trying to find patterns that are commonly there, right? So that looks a lot like Hebbian learning. However, in deep convolutional neural networks, unsupervised learning is artificial networks. Unsupervised learning is very rarely used for a number of reasons, but primarily because it's too hard. It relies on too much experience of the world to learn. So supervised learning is just faster and more efficient, right? But it is likely that, you know, as we start developing sort of, you know, sci-fi computer systems as they are now, that just like are exposed to the world and then just learn from experience of the world, that mechanisms like this would be used instead. Right? But we generally, now we're using it for engineering instead, right? So we're using it for finding images of cats on the internet. So we want it to happen faster than that. We can't just expose a network to the internet and say, you know, where's the cats? You know, it doesn't work like that, right? Supervised learning is much faster. Okay, so we've seen that the weights on filters primarily result from learning, but some filters. What's my point here? Okay, yeah, all right. Okay, so some filters appear to be hardwired, particularly in early stages of vision. So these appear to arise from evolution rather than from learning. All right. And this suggests that they provide a useful structure that subsequent filters can then build on. All right. So things like edge detectors. Right. So in artificial deep networks for vision, the filters in the first layer are often also hardwired edge detectors. And understanding these filters gives us an idea of which feature maps arise in biological vision and how they're used in subsequent stages. Okay. So understanding these early filters also gives us an idea of, is also important because pooling and normalization stages that we've ignored so far, okay, depend on the structure of feature maps. Right? So if we want to look at biological feature maps, and how these things work, we can't just look at an individual neuron. Right? We have to look at the activity of large populations of neurons working together. Right? So these mechanisms, feature maps, pooling, and normalization, are all related mechanisms, right, that don't just rely on one single neuron. So we're going to take a short break now, and when we come back, we're going to look at these processes in larger groups of neurons, okay, and how these larger structures do allow things like pooling and normalization. Okay. As I said just before the break, these last couple of operations, well, these last aspects, don't rely on one single neuron. So we have to move beyond looking at one neuron. And indeed, they don't rely on a single unit in a artificial network either. All right. So we're going to look at this using the example of the retina, so the very earliest stage of visual processing, and a little bit about the early visual cortex as well. So how are feature maps arranged in the input image? Right, so I mentioned in our last class that often we have the input image as being 3 color channels, all right? And so the convolutional filter, therefore, needs to include all 3 color channels, all right, to detect features at the first stage. Right? The eye also actually has 4 different sensors for light. 3 of these are also color channels for red, green, and blue, effectively, which are the 3 different cones, the color-sensitive photoreceptors in the eye. And the 4th one is a thing called the rod, which is not particularly color-sensitive, and it isn't used in color computations, so it's not really carrying color information to the brain. But it responds very quickly and under low light conditions, the rod. So this gives very good information for analyzing fast-moving objects, for example. So all of these 4 photoreceptors, so these respond directly to light falling on the retina. Okay, so they're the very, very first stage of visual processing. And they're essentially already 4 feature maps for 3 different colors and 1 or so for fast-moving, non-colored stuff. Right? So the S cone, which we see in blue here, responds to short wavelengths, so blue light. The M cone, which we see here in green, responds to medium wavelengths, and the L cone responds to long wavelengths such as red. Okay. And the rod responds to something in the middle, quite a broad range of wavelengths, and it responds very quickly and under low light conditions, which the other colour ones don't, which is why we don't have good colour vision in low light. Unlike the cameras that provide an input to an artificial network, these light sensors, these photoreceptors, are not evenly distributed across the image. All right? So a camera gives just as much detail in the whole image, but our vision doesn't work like that. All right? Cone density, so the colour photoreceptors, is much, much higher in the centre of vision, that is to say, where we're looking. All right? Rods, on the other hand, are much denser a little bit further out, okay, which gives us sort of good ability to detect moving things with our peripheral vision. Well, rods are a little bit more physically large, so you can cram in more cones in the centre of vision, which is what we're seeing here. But if we add these two curves together, right, we can see that the overall density of photoreceptors in the centre of vision, so what we're seeing, this location here, is very much higher than elsewhere. So vision in the eye strongly overemphasizes the center of vision, that is to say where we're looking. And the benefit of this is that it greatly reduces computational load on the brain, right, while it's still giving high detail in central vision. Right? So we have a lot of neurons responding to the center of the visual field, not so many where in our peripheral vision where we don't care about so much. So it gives us the ability to see things very accurately, right, but also cuts down on the processing that we have to do. Even at the retina, though, the brain's representation of visual space is flipped upside down and left to right. That's because the light rays from a particular position in an external image, right, need to pass through a little aperture here, much like we see in a camera, and therefore the light rays kind of cross over. Right? So the image on the retina gets flipped left to right, and indeed the image on the camera is also flipped left to right. It's just afterwards we flip it back again. This overrepresentation of central vision and this image inversion continue into the cortex, so into the early areas of the visual brain. Right. So here we can see that, you see this red circle in the middle. All of these circles have the same size, right? But on the retina, on the visual cortex, they're projections, so the amount of neurons responding to each of these circles is decreasing as we go from the center of the visual field to the peripheral visual field. Okay. We also see that everything's flipped upside down and left to right. So this is the right hemisphere of the brain, and it's responding to the left half of the visual field, and its representation of that is also flipped upside down. So you might think this strange representation of vision would cause problems for perception. However, the location of information on the retina or cortex is not important when we stop thinking about this activity as being an image, right? And instead we think of it as a set of feature maps or neural activations. Right? So that's a neural representation of image transformations rather than the image itself. As I said in the last class, it's very tempting to think of these sort of higher layers of neural networks as being an image, but they're really not, and it's a very limiting way to think about it. But what is very important here is that neighboring locations are, again, represented next to each other. All right? So places that are next to each other in the image are also next to each other on the cortex, even though everything is distorted to overrepresent the center of vision and flipped around as well. Okay. This is important when we consider that the spatial extent of a filter represents a continuous piece of an input image. So if we think about the tree of dendrites that we're looking at here that's going to interact with a group of neurons in a particular part of this image, right? That tree of dendrites only has a particular spread, all right? So it can only interact with a particular small part of the image, right? So keeping all of the neighboring locations in the image together in the neural representation, right, allows us to do computations that analyze spatial relationships in the image. Even though this isn't an image anymore, it's a neural activity pattern. So this is the main reason why the brain also maintains spatial relationships at each level of processing, which is again something that the deep convolutional neural networks imitate. All of the brain's sensory inputs actually similarly overrepresent the most important parts of the input. Here we can see the sensory and motor cortex, which are both organized as maps of the body. Right? So you can see a gradual progression where sort of nearby locations are represented next to each other. Okay. But we can also see here that certain parts of the body are strongly overemphasized. So here the face is much larger, right, than the leg, which is actually a larger structure. Right? Here we can see the torso here, trunk, is much, much smaller than the face and the hands in the brain's representation, although it's very much larger on the body. So this gives us a very detailed representation of the sensitive parts of our body, which also tend to be the dexterous parts of our body, the ones where we can perform fine movements. Similarly, if we look at the auditory cortex, which represents things by the frequency of sounds, okay, auditory frequencies are more, vocal frequencies are more heavily represented than other frequencies, because for humans, vocal frequencies are very important. Artificial sensory systems generally don't use such distorted inputs and instead aim to process everything in the same great level of detail. All right? This has advantages and disadvantages. The main disadvantage is that processing the whole image in a similar level of detail is quite computationally intensive. Right? So that requires a lot of processing, whereas just concentrating on the important parts requires less. The main advantage, though, is that cameras that provide the inputs to artificial networks don't need to move to resample important parts of the image. Okay? Our eyes move around to pick out the important parts of an image, right? So, but we can only look at one place at a time. Right? But normally, the inputs to an artificial network are static images, right? So moving the camera around the image isn't possible. Okay? And indeed, it will take considerable advances in deep learning before they can process images immediately, decide where the important details are, and move the sensors to sample those details. Right? Yeah?
Isn't it a different problem that you're trying to solve? Because the convolutional neural network is really trying to process do the processing of an image, whereas as humans, we're more the reinforcement learning kind of thing, where we want to have a goal, we want to do something, and then use that process.
Yeah, right. Indeed, they are slightly different things, but again, if you have a deep convolutional neural network, right, an artificial one, then what a human would do is it would look at the object. In the example of object recognition, right, a human would look at the object, right, and so have a finer representation of that object, right, and ideally, artificial network would do the same thing. Right? It would have a finer representation of the object that it's interested in. But it doesn't know what object it's interested in, whereas a human does. Yeah. So you're right, they are kind of different problems, but there are also different limitations in the two systems. Okay. So the first filtering stage in the eye analyzes the spatial relationships between nearby locations. Okay? And it simultaneously analyzes the relationships between different color maps. Effectively, then, these are filters with both the spatial spread and also a spread across the 4 different color feature maps. Okay? So we're going to look at these two separately, all right? And we're going to look at the spatial aspect first. Right? But actually, they're both happening together, which will become clear in a minute. All right? So the retina has several stages between the photoreceptors, which are what we're seeing here, which tile the retina. Okay, and the retinal ganglion cell, which carries the response up to the brain. The first of these stages compares one group of photoreceptors with the surrounding group of photoreceptors, right? So that the center of this group, right, is going to cause activation of this cell, okay, whereas the surrounding areas will cause inhibition of this cell. Okay? And then that will determine the firing rate of the downstream cells. So expressed as a convolutional filter, this might look something a bit like this. Okay? So here we've got a excitatory center, this group of positive numbers here with positive weights on them, and we've got an inhibitory surround with a particular spatial extent, right, which has negative weights on them. Okay. Now, if you were to if you were to activate this whole filter, okay, just like if you were to activate this whole group of photoreceptors here, then the actual result of activating all of these pixels is 0. Okay, so summing together all of these pixels just happens to make 0. I've designed it that way. Okay. And so if we then have a white light on the retina, okay, then this cell won't respond at all. If we have a dark spot on the retina, a bright spot on the retina just at this location, then this cell will respond very strongly. And if we have a bar going across across this little patch of photoreceptors here, right, then it will produce a positive response, okay, but it will be weaker than if it's just a single point. Just like a convolutional filter, there are overlapping copies of cells like this here as well, and here as well, and here as well. So each of these photoreceptors is actually synapsing with several horizontal cells that are kind of shifting this filter around. All right? So they're all kind of expressing a similar filter, if we want to think of it that way, but all in parallel, right? And all at overlapping locations. Photoreceptor that falls in the negative zone of one horizontal cell filter here will fall in the positive zone of another. A complementary filter is also present. So here we see that the center is positive, right? So that will respond to light, and the surrounding area here is negative. So this will respond most when there's light here and darkness here. Okay. But a complementary filter will respond to no light here and lots of light here. Okay. So that responds to darkness basically. This will respond to a dark spot here. So this is called an off-center response, whereas the previous one is an on-center response. Okay. So that here we'll see, if we see kind of positive as being bright in this case, all right, then this cell will respond strongly when there's a dark spot in the center and light around it. Okay. Oh, I missed some minus ones at the top there. Anyway, yeah, so that's going to then signal dark features, all right, and you know, we tend to think of light as being a positive thing, right? But what you're doing right now is you're looking at a light field where the features that you're interested in are defined by darkness, right? So this is text, right? So this is black text on a white background. We don't care about the white background. We do care about the black text, right? So it's just as important to kind of have the opposite filter as well. Right? So yeah, that's just a little detail here. So this set of on-center and off-center responses, therefore, effectively form two separate feature maps for lightness and darkness. Okay, and each of that is happening separately for all of the three color channels. All right. So the area that produces the positive response here is known as the receptive field of this cell. Okay. The term receptive field, right, describes an area in an image in biological systems. In artificial neural networks, the same term, receptive field, is often used to describe the spatial extent of a convolutional filter operating at higher layers as well. All right? So in an artificial neural network, a receptive field can mean the area that a convolutional filter analyzes in the previous network layer, not necessarily in the image. Right? In the context of biological vision, it's always referring to a position in the image. Right? And this is partly because it's very difficult to determine the spatial spread of a cell's inputs in a previous neural layer in a biological system. It's a very difficult practical experiment to do. right? So we always define everything in biological systems in terms of the actual image because we know exactly what we showed to the eye, right? Whereas in an artificial neural network, it's very easy to analyze the activity at the previous layer. In biological systems, there are methods, and we'll look at some of them in the next lecture, to look at how one network layer samples from the previous network layer. But in this case, we're talking about a receptive field as being in a particular part of an image. Right? Question?
I'm missing how you can respond to something that's not there in the biological system.
Yeah. Right. So these cells have a baseline firing rate, so they're always firing, right?
Oh, okay.
Yeah. So that firing actually specifically. Photoreceptors are the one type of cell that don't actually fire. They have a continuous graded release of neurotransmitter, and that can go up or down. Okay. So if we look at, just sort of to highlight what we're talking about here, right? If we get an on-center ganglion cell that will respond to light in the center, right, and we show it darkness, it will have a low baseline firing rate. Okay. The strongest response will happen when we show light in the center and darkness in the surround. Okay, see here. So this is basically responding to contrast, right, between the center and the surround. If we activate the whole area here, we see an initial response when the stimulus turns on. So here we see the stimulus turning on and then turning off here. Okay. We'll see an initial strong response when the stimulus turns on, which is signaling contrast in time, right? And this is basically because these negative connections, these sort of surround connections, happen a little bit more slowly than the center connection. So there is an initial brief response, but then quickly, while the stimulus stays on, right, the firing rate drops back down to baseline. If we show the kind of anti-preferred stimulus, that is to say, dark in the center and bright in the surround, this will inhibit the cell's response below baseline. Right? And when the stimulus turns off, just kind of as a contrast to this, so we kind of see the inverse thing, when the cell turns off, we kind of are signaling a change to the previous stimulus state, and so the cell will fire. All right. An off-center ganglion cell is basically doing the opposite thing. All right, so where we're showing nothing, dark everywhere, it will have a low baseline firing rate. Okay. Its preferred stimulus is dark in the center and light in the surround. So it will respond very strongly when we show that. Okay. It will respond initially when we show light in both the center and the surround. Okay. And it will be inhibited below baseline if we're showing the anti-preferred stimulus, that is light in the center and dark in the surround. Okay. So all of this is kind of relevant for understanding the relationship to deep convolutional neural networks, because as a result of this surround suppression, the response to light intensity inside the receptive field is normalized by the light intensity outside the receptive field. Okay? So the response to light in the center is kind of dialed back down if there's also light in the surround as well. Okay? So the light level in the surround is effectively used to normalize the light level in the receptive field, yielding a measure of local contrast. All right? So this resembles the normalization operation in a deep network and effectively converts light levels into contrast levels very early in the system. So this is happening in the retina, the very first stage of processing. It converts the amount of light into the amount of contrast. Right? However, the mean light level used here is the mean light level over a specific small part of the image. This isn't taken over the whole image. All right? Whereas in artificial neural networks, right, the normalization is generally done over the whole feature map, not just a local part of it. Right? Also, in most cases, this mean light level is typically restricted to a single feature map, right? In biological systems, but sometimes normalization is done across different feature maps in artificial neural networks. So here we're looking at local normalization rather than a sort of whole image normalization. Right? Now it's unclear whether these differences are better or worse than global normalization. They're just different. One thing that they do allow is they allow more complex patterns of normalization, which might be useful if an image has light parts and dark parts. Effectively, it increases the dynamic range of the representation of contrast compared to just normalizing everything over the whole image. Much like a high dynamic range camera sort of sort of adjust the aperture for different parts of the image. Right? Anyway. Okay. Do you have a question?
Yeah, sort of. Isn't this just the rescaling of the input? It's not really about the normalization you find.
It's quite different because it's local, yeah. So it's not a global normalization. There isn't really a global normalization of biological systems.
No, but I mean, like, usually you have like Vesper somewhere.
Yeah.
Where you rescale based on the output between the variables, etc. But that's not really what we're seeing right now, right? Because usually you also rescale the input. That's more like this.
Yeah, it is a bit like this. Yeah, yeah. So it's a related process, but as I say, it's quite different. Yeah.
Yeah, but then I'm missing the normalization.
Yeah, so it's just normalization at a local level instead of a global level. So it does something a bit different. Right. Okay. So this is so far we've just kind of limited everything to spatial. The spatial representation, and we haven't looked at how this representation changes across different feature maps. Okay. So together with this spatial comparison, there's also a comparison between the different color codes. Okay. So this forms 4 different feature maps, one for greenness, one for redness, one for blueness, and weirdly one for yellowness, which is strange because we don't have a yellow photoreceptor. Okay. To determine the amount, one important detail to notice here is that blue cones, short wavelength cones, are relatively rare in the human retina. Okay. So if we want to determine the amount of green, we ignore the amount of blue completely, right? And we just compare the amount of green to the amount of red in the image. So if there's a lot of green compared to the amount of red, right, then this output will signal green. Similarly, if there's a lot of red compared to the amount of green, so this is activated by the red input, inhibited by the green input, then the output will signal the amount of red. Okay. Yellow is calculated by being the sum of red and green. All right. So if we want to determine the amount of yellow in an image, right, this can be that will activate both the green and the red cones. Right? So the sum of the green and the red cones gives a good proxy for how much yellow there is in the image. Right? And yellow is kind of opposite to blue, so if the blue cone isn't activated, right, and the yellow and the red and green cones are activated, then that tells us that we're seeing yellow. And if the blue cone is activated and the red and green cones aren't activated, that tells us that we're seeing blue. We also determine whether we're seeing light and dark, which is simply all of these cones added together activating this output, right, for white, right, or light, probably bright is the better way to put that, and all of these are inhibiting the output for black. So at rest, if we have our eyes closed, this cell will be very strongly activated, right, because it has a high baseline activation, and then will be inhibited by any light coming in. All right. The main benefit of this is that it's approximately optimal for encoding, for representing color information. Right? So what we're doing in these comparisons is we're analysing the ratios of red versus green, the ratio of yellow versus blue, the ratio of light versus dark. All right? So using these ratios, each individual colour representation gives an accurate description of the colour of light falling on the retina, not just its intensity. So let's look at these 2 cases here. We've got 2 yellows here. One is a dim yellow and one is a bright yellow. If we're just looking at the activity of a single cone, so let's take the green cone in the middle here, right? Then this is going to say, all right, well, there's some yellow in the image, right? But if we're looking at the ratio of the activities of these 2 cones, the red and the green cones, we're saying, yes, there's a particular colour at this point in the image. And the ratio is also the same here if we make that colour more intense. Right? So here what we're doing is we're separating the activity of the cells, so that's basically the intensity of the light, from the balance of colour that is falling on the retina. Right? So here we move from encoding of the amount of red, green, and blue to the ratios of red, green, and blue, which is to say the actual colour of the light. So doing this comparison early in the retina, and indeed this is as early as it can be, it's really at the first synapse, means that all later processing can be colour sensitive, because we don't care about the relative amounts of red, green, and blue, and indeed white light has plenty of red, green, and blue in it, right? But we don't see it as red, green, and blue, even though the photoreceptors, all of those photoreceptors are activated. We see it as the ratio of red, green, and blue, which is they're all the same, they're all high, therefore it's white. Okay, so far we've treated spatial comparisons and colour comparisons separately, but actually they're done together. So the filters cross both colour and space. All right. In the case of the bistratified cell that we see at the bottom here, there's no suppressive surround. All right. There's no separation between a centre and a periphery. There's just a separation between blue light and yellow light, as this is here. Right? But if we look at these other cells, they have centres that respond to one colour, centres that respond to one colour or its absence, right? And surrounds that respond to a different mixture of colours or their absence. Right? So these filters are working both across the colour dimension and the spatial dimension as well. As we go from central vision, so that's where we're looking, to our peripheral vision, higher eccentricities here, the spatial extent of these integrating filters increases. So that is to say, the size of the horizontal cells and the retinal ganglion cells increases. The spread of the dendritic tree increases. So unlike in artificial deep networks, filters aren't the same over the whole image. All right? They go from being very small in the centre of the image, where we're looking, to being much larger in the peripheral image. Okay. There are also, even within a particular single eccentricity, right, there's quite a range of receptive field sizes. So that is to say, there's quite a range of different filter extents, even at the same retinal location. All right. So this means that basically we're not using a filter of one size, all right, to analyse the image. We're using filters of multiple different sizes simultaneously, all right, for the same part of the image, all overlapping, all having the same inputs, but doing, even at the first layer of processing, using different spatial extents. All right. So to produce the maximum response, then, we need to give an input that has both the right light and dark, so this is what we see here, light in the centre, dark in the surround, but also the right frequency, the right spatial frequency. Right? So here we can see a kind of medium-sized wave, right? And the centre part here matches well with the size of the wave. So the size of the centre matches well with the size of the wave. The size of the surround also matches well with the size of the wave. So that will cause this cell to respond very strongly. Right? But if we have a much coarser grating here, right, then when we activate the centre, we're also activating the surround to some extent, so the firing rate is less. And if we have a very fine grating here, we're activating the centre a little bit more than the surround, but not very much more than the surround. So again, the response is less. So to produce to produce a big response, right, then the size of the filter needs to match the size of the features in the image. Right? And because we have different filter sizes, okay, we can see that this relatively large filter size, which has a larger centre and a larger spatial surround as well, okay, will match stimulus like this much better, whereas this filter, which has a much smaller centre, right, will match features like this better. And all of those are happening in parallel. Okay, so using these different sized filters, these different sized receptive fields, the retina can break down any image into different components at different spatial frequencies. Right? So if we take an image like this, all right, these different components have different properties. All right, so here we can see three different transformations of this image. Okay. So at low frequencies, which we see at the top here, we see the global structure of which parts of the image are light and which parts are dark, right? But we don't see where the edges are. So we don't have a fine representation of where the edges are. Whereas at high spatial frequencies, we see where the edges are, but we don't see which parts of the image are light and which parts are dark. But if we keep these different parts, right, we then have all the information to reconstruct the original image. Right. So these representations at different spatial frequencies effectively also form different feature maps, even at the first stage of processing. This is quite different from what we saw in artificial deep networks, where each layer is convolved with filters of a single spatial size. In artificial networks, larger scale features can only be detected by features at later stages after intermediate stages of spatial integration and pooling have happened. So, if we look at retinal feature maps then, just to summarize what we've been looking at, in the input image from the photoreceptor, we have a separation of red light intensity, green light intensity, and blue light intensity in the three different tones, and we have overall light intensity represented by the rods. By the retinal ganglion cell, which is a very, very early layer of processing, this has been expanded out considerably. We then have separate feature maps for redness, greenness, and blueness, yellowness, lightness, and darkness, right, which are all calculated from these inputs. We have many different spatial frequency ranges, right, and we also have lots of combinations of these, of mixtures of color and spatial frequency. Right. So these filters in the retina perform a range of comparison operations over their inputs. Okay. So if we look at normalization and pooling in these contexts, it'll be about 5 minutes. Normalization is effectively the inhibition of activity by average nearby activity. Right. Pooling, well, in biological systems, there isn't a distinct problem of computational load. All right? So we don't have computational load, everything just works in parallel in the brain. But progressive layers do have smaller representations, as we'll see in the next class. As you go up from early visual areas to later visual areas, the representations of the visual field get smaller and smaller, but there are more and more of them. So progressive layers do have smaller representations and they do have more feature maps. Okay? This just doesn't rely on a specific pooling stage. All we have instead is we just use fewer neurons to sample the previous layer, okay, as we go higher and higher up the network. Okay. So we looked at shared weights before, right? So in biological systems, we don't have shared weights. Right? Weights from each synapse develop independently from other neurons. So there's no fixed weights that are spread across an image. So this is quite different from what we saw in artificial networks. There's no fixed filter extent at the retina. Okay? Receptive field sizes, that is, say, filter sizes, vary with eccentricity, so from central vision to peripheral vision. There are also multiple receptive field sizes at the same location. Right? So there isn't even a fixed extent of the filter across the image. And there is a tendency for the filters to be smaller in central vision and larger in peripheral vision. Right. But if a filter structure develops at one part of the feature map to compute something useful, similar filter structures are likely to develop elsewhere as well. Okay. So shared weights aren't built into the system, but it may happen that similar filters develop across the whole image because something that's useful in one part of the image is also useful elsewhere in the image. Okay. Right. So remember that a major benefit of shared weights is that convolution is a fast matrix operation, but it relies then on having a single filter. This limitation doesn't apply to if filters are made from distinct biological elements. We're not doing matrix multiplications here. There's no convolution operation. There's no single processor core that needs to efficiently process the operations of all of these units. Another benefit of shared weights that we saw was that the target of machine learning is very small if a small number of filters are used, therefore the same filter is used everywhere. Okay. Again, this isn't important if the filters are made from distinct biological elements. Hebbian learning, unlike backpropagation, operates independently at every synapse. So it's quite a different mechanism, and therefore the result comes out quite different. All right. Indeed, these differences in object recognition abilities across the visual image are actually quite clear in perception. It's just that we're quite able to ignore them because we move our eyes well. Okay. So here we can see a couple of faces of famous people. All right, you can identify them very clearly if you look at them. But try identifying these faces with your peripheral vision by looking here. Okay. So we're going to look here, and I'm going to start something happening. All right. Now try to identify these faces using your peripheral vision. All right. So if you move your gaze between the cross and the faces, we can see how the faces appear with and without high spatial frequency information. And this shows that even at higher levels of object recognition, the brain's features for visual information are not the same over the whole image. We're not sensitive to the same features over the whole image. So, and part of the reason why this happens is because we never have to recognize faces using our peripheral vision. We always look at faces, so we don't need to do that. So those filters have never developed, right? So we can see that actually there are perceptual consequences to the lack of shared weights here. Right? So we have different filters across the image, and so things look different across the image. Okay. So to summarize what we've looked at today, all right, biological networks share most operations with artificial networks, except pooling, but typically there are some differences between them. The filter and convolve operation primarily relies on dendritic tree synapses interacting with many upstream neurons in the previous layer. Okay. Different synapses have different strengths, okay, much like the elements in a filter. Okay. And filters cross multiple feature maps, like the different colors, even at the very earliest stages. Right? And that does typically happen in artificial neural networks, particularly where the first stage is a color image. The threshold operation relies on activation of voltage-gated ion channels, following the summation of excitatory and inhibitory postsynaptic potentials, and that leads to action potentials, that is to say, activation of the postsynaptic cell. The normalization operation indeed occurs quite differently. This is inhibition of activity by local mean activity rather than global mean activity. And learning relies on Hebbian learning, that is to say, cells that fire together typically wire together. So this strengthens responses to patterns of activity that have previously caused responses. Okay. So just to summarize what the major differences are, though, feature maps don't represent the whole, in biological systems, feature maps don't represent the whole image in the same detail. There's a strong bias towards the more important or sensitive parts of the input, in the case of vision, central vision. So there are sensitive areas that in biological systems that can move to sample task-relevant information. So we can move our eyes to do the things that we want to do. We can move our hands to touch the things that we want to touch. So far, artificial deep networks haven't got that. Okay. In biological systems, filters do have a limited spatial extent because dendrites can only grow so long, but they don't have a fixed extent. Right? So there are multiple spatial scales analysed at the same layer. In all of these higher level feature maps, though, image layout, spatial relationships are maintained in later stages, allowing analysis of spatial relationships at many levels. And this is like the fundamental thing that deep convolutional neural networks do, and biological neural networks are doing something pretty similar, at least in the case of vision. In biological networks, filter weights are not shared across each layer. Okay, we don't have shared weights, but similar filters are likely to emerge in many places. In biological systems, the maximum response is always part of the threshold. There's always a maximum firing rate, though it isn't always necessarily reached. And finally, we saw that normalization depends on local rather than global mean activity. And learning is generally unsupervised rather than supervised in biological systems. Okay. So there's plenty of similarities, plenty of parallels here, but also a lot of differences that make these two systems somewhat different. Okay. That said, deep convolutional neural networks are still the closest thing we have to doing anything like biological systems do, because they include several important aspects of biological systems. Okay. All right. So that's it for the lecture today. What we're going to do for the rest of the class is we're going to start on lab assignment 1. Okay. So this assignment, the aim here is to build some simple deep networks for object recognition in images. So this is the same kind of example that we're using for our biological systems and our artificial systems here. Okay. We're going to do this using a modern high-level neural network library called Keras. And the great thing about Keras is that it makes it super easy to describe neural network layers and neural network operations. Okay. A neural network layer is typically described in Keras by one line of code, and it's a really simple line of code. Okay. So it makes it super, super easy to write neural network structures. So the functions used are very straightforward and they're very intuitive, and they describe neural network layers and operations that we've been talking about so far. I suggest, and I really strongly suggest, addressing this through R and RStudio, and this is partly for consistency with other courses. All right. However, so particularly if you've taken cognitive modelling, you're using R there. Right, and so the things that you've learned there can therefore kind of be used here. RStudio in this case is relying on TensorFlow. Well, Keras is relying on libraries in TensorFlow, which is written in Python. So you need to start by installing these libraries. And R is primarily addressing, as the start of the assignment says, R is addressing Keras, which is addressing TensorFlow, which is addressing Python. So you need all of that stuff installed. If you don't know R, right, you've got a couple of options. There's one that I recommend, which is R's really easy to write. So I recommend going through this crash course that we have in the assignment section. This just teaches you the basic commands in R. It's super easy. So if you can program in anything, this will only take you a few minutes, and then you'll know how R works. Okay. Another option is to do this in Python, if you're really good with Python. However, the assignment contains blocks of code that are written in R. Okay? So if you want to do that in Python, you're going to have to translate those blocks of code into Python, which is going to be relatively challenging. However, if you are a really good programmer, that will be, the rest of the exercise will probably be really easy for you. So yeah, translating it into Python might be a more challenging thing to do. So if you think you're a really good programmer in Python, you might choose to do that. Okay. Normally, you're going to work in pairs. So you're welcome to work alone, but most of you will work better in pairs. But if you are working in pairs, you should both do the exercises on your own computer together. All right? So you shouldn't just work on one computer, you should work on two computers together. All right? You can share what you're writing, using Google Docs or whatever. But the point of both of you doing this, right, is that it greatly reduces the errors that you make. Because if you're both writing code, you generally won't both make the same stupid mistake. Right? And it also greatly increases what you'll learn from doing this. Right? So this lab, just to give you a bit of an idea, it varies hugely how long it takes between people. But it varies in quite a deliberate way. So there are parts of this that rely on quite a lot of sort of cognitive knowledge, and parts of it that rely on quite a lot of programming ability. All right? And we generally find that people who can do one of those things well need some more help on the other. All right? So generally it takes people about the same amount of time. If you are a really good programmer, you will find that the programming part's really easy, but then you should concentrate on the other part, which might be harder for you. Right? So don't just think this is easy because I can program it easily. There's lots of parts that are more challenging in there because they're not just programming things. So the amount of time it takes varies a great deal between people, particularly the amount of time the coding takes. It normally takes you somewhere between 12 and 15 hours. We've got 10 hours of class time. Help is available during class time. All right. So where we have class labs, you can come and get help. But you don't necessarily have to do this in class time. You can do it in your own time. You can schedule your own time. All right. The labs are really a way for you to come in and get help with it. All right. So that's really up to you how you tackle that. If you feel that you don't really need much help, you are probably going to do well by working on this without much help in class. If you need a lot of help, and particularly in installing the libraries that you need at the start, a lot of you will need a lot of help, then help is available during class time. All right. So we're going to stop there, and you can find all of the assignments in the assignment section of Blackboard. And for the rest of today's class until 1:45, you should be working on that. And I would say for a lot of you, a lot of today's class is going to be involved in making sure everything's installed and working, because that's not particularly easy for a lot of you. There are some notes about that in the assignment section as well. One thing that we have found a lot is that people have problems getting things working because they already have R or RStudio or Python installed, but they're not using the latest version, right? So the first thing you should do, if you have everything installed already, right, is update it to the latest version, all right, particularly if things don't work. Okay, so with that, I'm going to let you make a start on the assignment. We're going to have help with this from Martijn, who's sitting in the front here, from Krista, who's in the back there, and from Jesse, who's just sticking his head around the door here. Yeah, over to you.